Well, hello everyone, and welcome back. I'm Jack Rincon, Bridge to AI Training Recruitment Mentoring Program Manager, and I'm here with Dr. Pepe Ping. And we're pleased to continue our Bridge to AI lecture series for 2025 and 2026. We're excited to share another intellectually rigorous session with the consortium community beyond LLMs, neuro-symbolic approaches for language understanding in the biomedical domain. We're also excited to note that this is the first lecture in our new module on novel AI technology. Dr. Pepe Ping will have the pleasure of introducing our first lecturer for this module.
Hello, everyone. Thank you, Jack. It's our great pleasure to welcome today's speaker, Professor Mihai Surdeanu. He is a professor of computer science in College of Science at the University of Arizona, with joint appointments in linguistics and cognitive science. He will be presenting a lecture titled Beyond Large Language Models, Neuro-Symbolic Approaches for Language Understanding in the Biomedical Domain. We are truly honored to have you join us today. I'm also very privileged and honored to give a short introduction for Professor Surdeanu. He is a leading investigator in NLP, where his work sits at the boundary of machine learning, linguistics, and knowledge representation. At the University of Arizona, he has held a number of very distinguished positions. In addition to his faculty position at computer science, he's also hosting positions in linguistics and cognitive science. All these appointments reflecting the interdisciplinary nature and success of his scholarship. His research programs spans question answering, information extraction, and interpretable language technologies. All of that aim to not only rendering LLM to perform well, but to make certain that the reasoning process is transparent and accountable. In this state-of-the-art and rapid development of AI technologies, it is important for us to recognize that Professor Surdeanu has received several important awards. He is the ACL Outstanding Area Chair Award and the University of Arizona's Distinguished Scholar Award in Computer Science. And he plays a major leadership role in the NLP community nationally and globally, currently serving as the acting editor for TACL and senior area chair for ACL and EMNLP. We are so fortunate to have you and gave us guidance about the state-of-the-art status in the NLP area today. Back to you, Jack.
In today's session, we'll focus on two learning goals. First, we'll critically examine the strengths and limitations of neural architectures, especially large language models in comparison to neurosymbolic approaches for biomedical language understanding. This is increasingly important as healthcare and life sciences rely more heavily on AI systems that must be accurate, interoperable, and trustworthy in high-stakes settings. Second, we'll explore how to design and build symbolic and neuro-symbolic NLP systems that are faithful, pliable, and fast, meaning systems that respect domain knowledge, remain adaptable to new evidence, and operate effectively in real-world biomedical applications. These perspectives will help you think beyond black box models and towards hybrid approaches that better align with the needs of biomedical research and clinical decision making. With that, I'll hand it over to Dr. Surdeanu to take us deeper into this topic.
All right. Thank you, Pepe and Jack, for the very nice introduction. Let me start sharing my slides. All right, I'm moving in slideshow mode. People still see the slides? All right, good. I'll skip the title slide. Thanks to your very nice introduction. I'll start with the obvious. LLMs or large language models are everywhere and they are great, right? In fact, they are probably the biggest and most important development in the history of natural language processing or NLP. However, because they are so good, we tend to skip over and ignore their drawbacks, which I think are fundamental in medicine. So let's focus on those for a few minutes. But before we get to that, uh, like any technology, LLMs will follow the hype cycle. And my prediction is that we are right about here, right? We're over the peak of inflated expectations and we're beginning to fall into the trough of disillusionment. And we see sort of economical impacts here. So companies that invested heavily in AI have not seen the productivity boost that they expected. LLM companies are, functioning under heavy losses and so on and so forth. So I think we have to adjust a little bit. And I think this is due to multiple important limitations of LLMs. So I'll go through each one of these in turn and give you some examples that certainly are relevant and applicable to the biomedical domain. So they're not explainable, right? And what I mean by that, I'll give you a simple example from from a paper very nicely titled "LLMs do not say what they think," in the sense that the explanations you get from an LLM do not correlate with what they do internally. So here's an experiment that came from this paper. And in this paper, they ask an LLM if a certain question is reasonable, is plausible or implausible, right? So this is a question about soccer, and the LLM, using chain of thought or COT, answered correctly first that this sentence is plausible because this action is part of soccer, right? So what the authors did in the second part of this experiment, they provided some examples in context for the LLM where these are multiple choice questions where the correct answers provided in the context as examples were always option A. In other words, they biased the LLM towards option A. And when they asked the LLMs again if this is a plausible or implausible question, the answer was A because the LLM picked up the hints from the context. But then the problem was it made up a completely hallucinated explanation that has nothing to do with anything. Really, the only correct explanation would have been, from your examples, I can guess that the only correct choice is A, so I'm going with that. Instead, it made up an explanation that's not correlated with what's happening inside. So this clearly is bad in the biomedical space, right? You don't want to have explanations that are fake. This is a quote from Dave Gunning, who was a program manager for DARPA's Explainable AI program, basically discussing how explainability is important for the Department of Defense if they are to use this type of systems. And of course, this applies to many other domains, most importantly, medical. And the second drawback is LLMs are not pliable. What I mean by that is, what do you do when the LLM is incorrect, right? And at this point, you may say, well, but Mihai, we have prompt engineering. But prompt engineering is not controllable in itself either. So this is a figure from another paper where the authors have shown that just making sort of minor irrelevant changes in the prompt, the performance on a text classification task decreases from roughly 80% to 3%, right? So for example, if you look at the prompt in blue, here you have a white space after the colon. And here, when you get 3% performance, there's no white space after the colon. So really something that makes no impact on semantics, yet the performance basically drops to nearly zero, right? So prompt engineering really does not offer the amount of control that we think it does. Also, they are not efficient, right? So an average LLM today functions with around 1,000 to 10,000 watts, whereas the human brain functions with roughly 12 watts, which is a lot less than a regular light bulb. So an LLM consumes 3 to 4 orders of magnitude or 2 to 3 orders of magnitude more than the human brain. And this is clearly not good if you want to deploy this at scale, for example, analyzing the entire amount of literature in the biomedical space. And they're also not factual. I don't know if you've seen this example. So if you ask an LLM to generate an image with a clock showing whatever time you want, it's always going to generate 1010 because those are the images on the web for clocks, and those come from some marketing genius who decided that this clock resembles a smiley face, so it's much better to use this for marketing purposes, and this dominate the web, and the LLM is biased by the most common instance. This comes from the homework of one of my kids who was working on generating a figure for calculus, and the LLM made up one of my favorite new words, Dorktion, which of course doesn't mean anything. They don't really reason. We'll come back to this example, but in one of our research works, we tried to get LLMs to think, to come up with multi-hop causal mechanisms that explain how two things are connected. In this case, how stroke is connected to intestinal inflammation. And this is done by chaining together multiple causal interactions, right? So you see here 3 such interactions being chained together, where each interaction comes from a different paper. So when we provided the corresponding papers to the LLM, it hallucinated a mechanism that's not supported by these papers. It may be correct or not. I'm not a biologist to evaluate this, but it's not supported by the papers we provided. And clearly, again, you don't want an LLM to hallucinate causal explanations in your work. And later on in the talk, I'll give you an example about how we fix this. Okay, so if LLMs are not the complete solution, I want to emphasize complete solution. I still think LLMs are great, and they will be part of the solution for AI in whatever domain, but it can be the complete one. So if they're not the complete solution, what is? And my answer today is that we have to focus on neuro-symbolic approaches, which combine the generalization power of LLMs with the adaptability of symbolic systems, which are driven by declarative statements, which we understand as human beings and can modify as human beings. Okay, I will discuss 2 papers plus some unpublished work today. The first paper comes from the work we did in DARPA's Big Mechanism program, where we built a machine reading system that was applied to discover or explain cancer treatments. And this is a bigger work, but the core NLP team for this work was Marco, Gus, Enrique, and Wendy. Okay, so why cancer? I'm sort of preaching to the choir to this group, but since President Nixon's days, the US has invested over $200 billion in cancer research, and we haven't seen the expected outcome out of that. So this chart shows cancer death rates among women between 1930 and 2005. And you can see that it's not amazing, right? Some cancers are going down, but not all of them. And a very similar story appears for men. And why is that? So if you think about biology in general, as I'm sure you do, it's a very complex field, right? There are many, many things going on. And because of the complexity of the domain, each researcher focuses on a narrow slice of it. So the way I like to think about this is biology is a puzzle that has billions and billions of pieces. And these pieces have been broken down into many, many small parts, which are published in millions of papers. So this is a simple chart I generated before this talk, which shows that PubMed has indexed way over 1 million papers every year, right? And 90% of these papers are not cited, are never cited. So I tell our students that if you get 1 citation for your paper, you're already way ahead of the curve, right? And Isaac Asimov said it much nicer than I can several decades ago, that science is essentially a puzzle. And it may be that somebody has the solution already and is not even aware of the puzzle that it may solve, right? And this is certainly happening today. In library sciences, this problem is called undiscovered public knowledge, in the sense that the pieces of the puzzle are there, but the assembling of the puzzle is not done because it's too big. It's too much work, right? So here's an example from Swanson's paper from 1986. So he discovered the paper where the authors discover that dietary fish oil reduces blood viscosity. We know this by now, but at the time it was reasonably new. And blood viscosity, this is a different paper published by different people. Blood viscosity promotes Raynaud's disease. That's the disease that sort of reduces circulation in the capillaries, typically hands and feet. And these things were never put together at the time. And obviously, if you put these two together, which is what the authors did, what Swanson did, you now have a treatment for Raynaud's disease. So this is just an example. And what I'm doing with my work is essentially this problem at scale. And basically, the hypothesis is that if humans can process this much information, then machines can help and should help, right? So we need machine reading of millions of publications in order to discover new treatments. So what we've done in DARPA's Big Mechanism program, we propose a system for machine reading, and the system ingests a bunch of publications on the disease of interest, in this case, cancer in general. And we do machine reading to identify biochemical interactions, such as ubiquitination, regulations of those, et cetera, et cetera. Using these individual interactions, we build causal models of cells. And using these complex models, we can now reason over them, and we can predict new treatments. We can explain existing treatments by sort of discovering the causal mechanisms behind them. I'll give you an example shortly. Okay. What we observed in this program was that the typical way to do this using supervised machine learning does not apply here because A, there was no training data, and B, there was a DARPA evaluation coming within 6 months. So if you're familiar with DARPA programs, they tend to evaluate every team biannually. And if you don't do well, funding is cut. So you have to do well. So we had to move fast. And most importantly, beyond funding, this was in the disciplinary project between natural language processing team and cancer biologists. And with domain experts such as cancer biologists, you can't give them a bag of probabilities, so to speak, that they cannot understand and debug. They need to operate over things they understand. So the solution was symbolic models. I'll give you some examples soon. But the main advantages of symbolic models is that humans can understand the model. They're typically driven by rules or statements, which are humanly readable. And more importantly, humans can edit the model. If the model performs something that is incorrect, you can just go in there and change it. With an LLM, you can't do that, as we discussed a few minutes before. Or in other words, the way a former collaborator put it, Dane Freitag from SRI, models must be pliable. Okay, so I'm not going to bore you with too many technical details, but I want to take you through a walkthrough example of the system, what it does. So we start, this is actually a neuro-symbolic system because the first part is neural. And with the first part, we do syntactic analysis of text. So what you see here on top of the text is a directed graph which captures syntactic dependency. So, I'm not going to bore you with this, but just as a simple example, N sub means nominal subject. So we is the nominal subject of the verb hypothesize. C comp is a clausal complement. So the clause driven by enhances is the complement of hypothesize, and so on and so forth. Using this information, we extract entities of interest. And in our case, entities of interest are proteins, pronouns, which may point to other proteins, diseases, but other biological processes. Then we move on to simple event extractions, which operate directly over entities. So in this case, we have an expression event, which operates over the PTPN13 protein, and a phosphorylation, which works with a protein that's unknown at this point. Then we move on to nested events, which are events that operate on top of other events. So we see here that the expression event positively regulates the phosphorylation event. The problem with this is if you look at the larger context, the positive polarity here is actually wrong because there is a negation on front of it. It's the decreased expression enhances its phosphorylation. So really, the correct polarity would have been negative regulation. And we do that by looking at sort of negative polarities throughout the sentence. Then we also extract hedging because some statements are stronger than others, and we want to discard weak statements, such as we hypothesize that something is happening. That's not a very strong statement, right? You want to have a straight statement that's supported by experiments. And lastly, we do coreference resolution. In this case, we link the pronoun its with F in B1, which indeed is a protein. Okay, so this is sort of the big picture. And out of all these layers, these 3 layers are done using rules. And in fact, it's a total of only 154 rules, which is not a small number, but it's not a large number. This took roughly, in my sort of arbitrary estimation, roughly two person months to put together. So it's a good investment, but it's not a huge investment. And just to give you an example of what the rule is, a rule in general is a syntactic patterns that carry some semantic meaning. So in our context, suppose that I have a rule that says if I see the verb promotes or enhances, and the syntactic subject of this verb is a protein, and the syntactic object is a biochemical reaction, then really what I'm looking at is an upregulation event where the controller is the subject and the controlled thing is the object. Okay, so we have roughly 154 patterns like this. So this rule would match sentences like the one here, where a protein enhances a phosphorylation event. Okay, moving on. So this is a summary of this first system. So how well does it work? So I mentioned before that DARPA evaluates systems biannually. So I'm going to summarize some results from one of these evaluations. And what DARPA did in this evaluation, they evaluate the system on two axes. The x-axis is throughput, meaning how many papers, these are papers, a system can process in a given unit of time. I don't remember what the unit of time here was, but it was an hour or two, I think. And on the y-axis, you have precision, meaning how many of the statements extracted by these systems were accurate. And you see that you really want to be in the top right corner, right? You want to have high throughput and high precision. And our system is here. Team 4 is a combination of our system and Team 2. So our system is here. And what's interesting beyond the throughput is that the system has roughly the same precision as human experts. Right. Of course, human experts have much lower throughputs because this human is not going to read 700 papers in a couple of hours. Let's look at why this is the case. This was a surprising finding to see that systems work so precisely. And as we'll see, this happens because in some cases, machines are better than people, and in some other cases, humans are better than people. But on average, they come out about the same. So we analyzed the errors of the systems, and we found out that identifying event predicates, meaning verbs such as promotes, enables, inhibits, etc., reaches our system, and they're roughly the same. They're both pretty good. The error rate is 7 to 5%, so that's good. Identifying the arguments was a problem for the systems, and this is done because our system uses syntax, and sentences in biomedical publications tend to be very long and convoluted, so the syntax analyzer got confused many times, whereas humans could process these complicated systems much more easily. We'll see that even LLMs are confused by this in a further experiment. So machines are much worse at picking up arguments of events, like which protein enhances this other event. However, machines are much better at grounding entities, meaning if I find a protein name, which protein exactly am I talking about? This means linking this protein name string, to an entry in UniProt, for example. So we saw that machines are much, much better than humans for this task because this is a repetitive mechanical task where machines are consistent, whereas humans get tired and bored and sort of drop the ball on it. So overall, on average, if you average these numbers, performance is roughly the same. Okay, so fine. We now have some claim that machines can read papers at the same precision as humans, but at much higher throughput. But is it actually useful? So I'll show you an experiment that we've done with collaborators from Harvard Medical School at the time. They are now at Oregon Health Sciences University on the mutual exclusivity phenomenon. So, some of you may know what this phenomenon is, but for the others, I'll explain it. So assume that you have a kill switch for a certain cancer, which is Y in this figure, and you want the kill switch to be active. Now, this kill switch can be disabled by multiple pathways. And you see here the pathway that contains the blue protein and the pathway that contains the brown protein. And what people have observed in patient data is that these mutations are mutually exclusive because one is sufficient to disable the kill switch. You don't need both, right? So the best analogy I heard about this is that this is equivalent to a burglar breaking into a house, right? The burglar can break the door or the window, but they don't need to break both, right? One is sufficient. So why is this problem important? The problem is that if you block one pathway, the the cells mutate again and we'll find that the other pathway, right? So the disease comes back, as I'm sure you know about cancers. And so, in other words, correct therapy for such problems is to disable all the upstream mutations or the driver itself, right? So previously, our collaborators were linking patient data where they observed mutually exclusive pathways with causal mechanisms manually curated in the pathway commas database. And the authors of pathway commas estimated that it covers roughly 1% of the literature, which is clearly not enough. So what we've done in this collaboration, we expanded pathway commas with mechanisms extracted automatically from publications. And just to give you some hints about the data, pathway commas contain, at the time, 89,000 interactions. We extracted in just 2 weeks over 1 million. And even if you say we want to have reproducibility, so we want we want to keep interactions that appear at least 2 papers, it's still an order of magnitude more than what pathway commas did in over 10 years. So I'll show you some results for some cancers. And what you'll see in these results is what you see inside each box are the mutually exclusive pathways that drive, promote, or inhibit a driver that's outside the box. So check one would be the driver that's relevant for some disease. And inside the box, you have the mutually exclusive pathways. And what you'll see in yellow are the interactions that are discovered by the machine after adding our data. Right. And of course, these yellow interactions come from statements in publications, such as the ones you see here. Okay. So these are results for breast cancer, which is famously known for mutual exclusivity due to the BRCA1 and BRCA2 genes. And you see here, if you just sort of take a step back and look at the colors in the figure, there's a lot of yellow in this figure. These are all causal mechanisms that were added by our system, right? So in other words, we now have a much better understanding of this type of cancer thanks to machines. And we have similar results for 7 different types of cancer. This is glioblastoma, and I'll skip the others in the interest of time. But we now have causal mechanisms added to this type of explanations, but how many are plausible? So to do this, we worked with a domain expert in cancer researcher, and he found that 51 of the hypotheses that we manually analyzed here, out of those, 65% were correct if the interactions appeared in a single paper. And if you keep only interactions that appear in 2+ papers, almost 80% of those were actually correct. So clearly, it's not 100%, right? So I would say this is good news in the sense that domain experts are still very much needed. You still have a job as a cancer researcher, but you can speed up your research a lot. Instead of sort of fishing around for hypotheses to explore for new treatments, you can let the machine suggest likely treatments to you, and then you have a much smaller search space that you need to explore. Okay, so this was the first paper. And what we learned from this paper is that mostly symbolic systems can obtain state-of-the-art performance similar to human domain experts, are fast. I didn't discuss that, but our system processes a paper in roughly 5 seconds and are pliable. If the rule is wrong, you can just go in there and change it. In fact, we have a log of GitHub pull requests, which are basically corrections submitted by our collaborators who are cancer researchers. However, one problem with the system is that if those 154 rules match, life is good. It's all great. The problem is when they don't match, there is nothing that gets extracted. And the reason for that is that rules don't generalize very well in the sense that they need to match that syntactic structure exactly. Any slight variation will break the rule. Okay, so in the second paper, we'll address this limitation. The second paper was carried out at the University of Arizona by these 4 students. And I'll give you a simple example of the lack of generalization power of rules. Suppose that this is an open domain example. Suppose that you have an information extraction system that wants to extract founders of organizations. And the rule that we have here says, I need to match the verb founded, and the subject needs to be a person, and the object needs to be an organization. right? Very simple rule. So clearly this rule is going to match the first sentence. That's good. It doesn't match the third sentence, which is also good because this sentence is about something else. The problem is it doesn't match the sentence in the middle, which, yes, it has a different syntax. Instead of having the verb founded, we have the nominalized form of that founder. But really, semantically, it's the same content, right? It talks about Bill Gates being the founder of Microsoft. So what we've done in this paper is we flipped this not match to a match, right? So at the very high level, what we've done in this paper, instead of matching the rules syntactically, we train a system to match them semantically. So let's discuss how we did that. I'll show you some results on this task, which is a very complicated task that's very much unsolved. This is called few-shot relation extraction. And in this task at a high level, you have to extract relations that appear between two named entities in a sentence. So, for example, in the sentence at the top, we have to extract the fact that Billy Mays died in Tampa, Florida, right? To make things even worse, this is done without training data. And at testing time, you have only 1 or 5 examples for the relations you have to extract. And the relations you have to extract are different than what you may have seen before. All right, so for example, at testing time, this is the first time we see city of death, and we only have 1 or 5 example sentences for this relation, right? So it's a complicated task, and performance, as you'll see, is not great. So the intuition for our system is we still start with syntactic rules. We call them hard rules because they need to match exactly the syntactic structure. But instead of matching them syntactically, we match them with a soft matcher, which looks at the semantic meaning behind the rules. Okay, so let's discuss these 2 components. The first one is symbolic and pliable. The second one is neural, but it's domain independent. You only have to train it once, regardless of the task you're addressing. So for the first one, suppose we're given 1 example per relation. We again want to extract founders of organizations. From this sentence, we use a syntactic analyzer to extract the syntactic dependencies. And we find the shortest path in this graph that contains the syntactic dependencies, right? So that becomes the rule. And you see here that the rule says that if I see the verb founder, a nominal modifier driven by the preposition of, which is an organization, and the subject is again a person, then I have a founder relation. Okay, so now we have 1 rule. Now we have to work on generalizing the rules such that it matches semantically. And this is the job of the soft matcher. And the key observation for the soft matcher is that you want to train it on pairs of rule, like the rule we just seen, with sentences that have a different syntactic structure, but the same semantic meaning, right? So that will help the system generalize from syntax to semantics. Further, this data set must be domain independent, because we want to train this just once and then apply it to whatever domain we're trying to solve. So the way we addressed this problem was we randomly picked up sentences that contain named entities. In here, you see sentences with person and organizations, but in the biomedical space, you can imagine sentences containing 2 proteins, right? So at this point, we don't know what relations may or may not hold. But we know that these entities are connected in some way. Using the same shortest path in the syntactic graph algorithm, we find the rule that holds. And then we feed the original sentence to an LLM to paraphrase it, right? So this is very minor usage of LLMs because LLMs have been trained on paraphrase data, so they're actually very good at this. In fact, in this problem, we tried very hard to find mistakes, but GPT for something, which was the GPT that was the latest model at the time, worked perfectly. We couldn't find a paraphrasing mistake. So what this means is that we now have rules that are driven by a certain syntactic structure aligned with sentences that have a different syntactic structure, but the same semantics. And then we just pair them together and train a classifier to align rules with sentences that are paraphrases of the original sentence and move them far apart from sentences that are not. So how well does this work? So I'll just give you a hint about, let's look at the second example. So suppose we have the sentence that we're trying to classify. John got his degree from Oxford. So we want to look for universities attended, right? And our rule simply says that person graduates from organization, in this case, university. And our system, our soft matcher says that this matches with 82% confidence, which is pretty good. So it kind of works. So let's see how this works quantitatively on all the data. So this is a very simple baseline that the student put together, which was, I'm going to assign the relations that has the same entity types. This is unsupervised. It's very dumb. It's very simplistic. And you see that this works at 10% F1 score, where F1 is an average of precision and recall. Okay. These are actually state-of-the-art systems in the NLP space, which are supervised systems that learn complex representations of the data in order to solve the task. And you see that in some cases, they're actually worse than the baseline. And even when they are better than the baseline, they're not much better. This is another system that was developed in our lab. This is similar to GitHub Copilot, which is that LLM that generates code. But in this case, we train it to generate rules instead of code. And you see that this one is better, but it's not fantastic. The next one is an LLM within context learning. This is about the same. So despite the fact that this LLM is much, much larger, it's still not great. Now, this is just the system that has the hard rules. And this is still not great. But once you combine the hard rules with the soft matcher, performance just goes through the roof. Right. So we have double the performance of the baseline, which I would say is very good. We still are far from solving this task. This is a very hard problem, and even a very large LLM is not much better than this. Okay. And our model is much, much smaller than any LLM, really. Okay, so one question, I will try to wrap up in about 5 minutes, but I just want to mention a couple of things first. One question that the critical reviewer may ask is, now that you're not completely a symbolic system, right? So we know that a symbolic system is pliable, but now you combine a symbolic system with a neural system. Are they still pliable, right? So in order to see if they are pliable, we ask two domain experts to modify the hard rules without modifying the soft matcher. And I'll skip some of the details, but the performance increases significantly, even though the neural system was never touched, right? So in other words, the pliability of the symbolic component permeates through the neural component to the overall system. Okay, clearly this is impossible to do with any neural network today. So what we learned so far is that neuro-symbolic systems are explainable, pliable, fast, and they do actually perform better than purely neural methods. Okay, so at this point, if you're a critical participant in this lecture, you may ask, is Mihai just a Luddite that hates LLMs and he never wants to use LLMs? So to show you that that's not the case, I will show you some work that's not yet published that was carried by Saiful and Harris, where we try to see how this type of symbolic knowledge can improve the LLM reasoning in the biomedical domain. So the task we design here is we provide an LLM paragraphs from papers. So you see here 2 paragraphs. Well, they're sort of summarized here. And then we ask the LLM a bunch of questions related to causal reasoning. So for example, such questions are, is ginsenoside RG1 causally connected to opportunistic infections? The answer, if you read this text correctly, is yes. How many causal hubs are between these 2 entities? The number is 2. They are connected through CD4 T cells. What's the polarity of the causal mechanism? And the polarity is in inhibition in here, right? So these are the questions we ask the LLMs. And what we've done to see if symbolic information helps was we provide more and more symbolic hints to the LLM from essentially nothing. In other words, here we just provided the paragraphs from the paper. Then we provided just the paragraph that contains the interactions where we know the causal interactions are. In here, we provided, I believe, ±1 paragraph. So a larger context that includes the correct information, but it's larger. In here, we provided just the paragraph where the correct information exists. In here, we did the same thing at sentence level. We looked at where the correct causal information exists in the sentence, and we provided ±1 sentences around it. In the next one, we provided just the single sentence where the symbolic system identified the causal statement. And lastly, the most hints are single sentences with markup, where we introduce some markup to indicate what the LLM thinks, what the symbolic system thinks is happening. So to give you just example of the last one, for the previous example, the symbolic system identified the causal statement driven by the verb increase between the entity in yellow and the entity in blue. In the second paragraph, the symbolic system identified lead to as a causal trigger connecting the entity in yellow with the entity in blue. So we just added markup to the raw text to indicate that this is what's happening. And this is an evaluation of many LLMs in here. And what you see here, by the way, all these are guided just through prompting. We provided examples in the prompt and a very detailed prompt telling the LLMs what to do. And let's just, in the interest of time, focus on no hints from the symbolic system, which is the bar in red, to all the hints, all the markup, which is the bar in green. So you see here that overall, let's just look at the average column. Performance more than doubles, regardless of the type of LLMs. These are LLMs that were trained for reasoning, and these are LLMs that were not trained for reasoning. And performance really doubles between these two configurations, which is really very strong proof that symbolic information can help keep an LLM on track. And just to give you hints for the next talk, if there will be a next talk, we have initial evidence that reinforcement learning improves performance even further, where you keep fine-tuning the LLM with this type of symbolic information. Remember that in the previous slide, we did not fine-tune anything. We just used prompting information. And further, you can map the symbolic information to specific neurons in the LLM. So you can imagine having your favorite protein mapped to a specific neuron in the LLM, which means you can explain when the LLM is thinking about that protein, and you can control it. You can force it to think about that protein. And if you're curious, this is done using sparse autoencoders coupled with a specific training method to do this type of alignment. I'm happy to talk about this offline. Before I conclude, I'll just conclude with one example here from our experiment. So you see that this is what I showed at the beginning. The LLMs where you just provide the raw text tend to hallucinate information. But when we provide these symbolic hints, you see that the LLM stays very close to the symbolic information. And it's actually a really good summary of what the symbolic system produced. Okay, conclusions. I would say LLMs are great, but their drawbacks definitely limit their applicability in critical domains. Neuro-symbolic architectures can address many of these limitations, and they are fast, explainable, perform well, and you can change them when they do not. And they can be used, as we saw in the last experiment, to successfully augment LLMs as well. All right, I think that's it. So I'll stop here. Thank you for your attention.
What a fantastic lecture, Dr. Surdeanu. Thank you.
Thank you.
I am going to invite my partner and chair of computer science at UCLA, Professor Wei Wang, to host the next 10 to 15 minutes question and answer session. Professor Wang?
Yes, thank you. I really enjoyed the talk. Thank you so much for sharing this with us. So we do have some questions. So everyone in the audience, please use Q&A to put your questions. So we have one at the moment. So is there a way to remove the human component in providing symbolic information? I.e., a large LLM can provide symbolic information and the sentence pairs, which can act as training data to fine-tune a smaller LLM.
That is certainly possible. There's no question about it that it's possible. In fact, we have seen in the past that you can ask an LLM to generate some of the rules that we use, for example, in the first paper. So you can, but the performance is never exactly the same. So this is an example of that. Let me just jump to some of the results in here. So this is in here, this is a system where we remove completely the symbolic component, and we asked an LLM to generate symbolic information and apply it. And you can see that, performance is better than many other purely neural methods, right? The previous two columns. But it's not as good as when you let people provide examples, right? And in here, in our system, we don't really write the rules. The rules are extracted automatically from some examples that people provide. And I would say that's good. I still believe that sort of hybrid approaches that combine domain expertise from people with machines are, at least in the short to medium term, the solution, because AIs are still sort of fuzzy, incomplete, noisy, etc. So keeping them on track is a good idea.
Yeah, could you talk about the criteria that was used to a certain plausibility when the plausibility analysis was made? Yeah.
That's a very good question. And again, I'm not a biomedical expert, so we defer to our cancer researcher. And basically, what we left it a little bit soft. And what we did was we asked him, when you find the sort of causal mechanisms that he investigated, and he read the papers behind them, did he think that sort of concluding result, connecting A to B and B to C, did he think that connecting A to C is reasonable? And that means analyzing the paper, seeing that the experiments are solid, and seeing that sort of this type of multi-hop causal connections make sense for his domain expertise. So we left it up to the domain expert. And of course, plausibility doesn't mean correctness, right? So as we all know, this needs to be validated in wet experiments, right? So that's why we sort of call this plausibility, because we have no evidence from wet experiments that didn't exist at the time for this hypothesis to see if they're actually correct. So in other words, let me rephrase this. Plausibility in this case was, would you try to design a wet experiment for this idea if it came to you? And if you said, yes, this looks like a good idea, then we call it a success.
Thank you. Well, this next question is for this last work regarding the video neurons responsible for particular concepts. Is there a notion of generalizability, the generalization of a concept that map neatly onto the neural network architecture? For example, do you see structures that correspond to the verb effects that encompasses the neurons for increases and decreases?
This is a great question. In fact, we're in the process of writing a proposal that discusses and proposes some solution for this. At this point, the work that you see here, which is under submission at the moment, but it's also on archive if you want to look it up, and there's a long discussion about this. We're basically starting with the assumption that the ontology of concepts we're trying to include is predefined, right? So this is a limitation, right? In other words, we have to know ahead of time what is the deterministic knowledge that we want to include. So in this figure, you see a very simple example where we want to include 6 types of relations: birth date, birth city, etc., etc. So if this ontology is defined ahead of time, then you can define a loss. We called it a binding loss. You see it in this figure, that basically uses cross entropy to link these concepts to specific neurons which are defined ahead of time. Right? So if this loss is trained, then the mapping is nearly perfect. It works. What you see here in the right-hand side of the figure, you see the diagonal matrix, where rows are the concepts, the symbolic concepts we're trying to learn, and the columns are the neurons that they are assigned to. And you see that the diagonal is almost perfect, which means that this binding loss is doing its job to bind these concepts to a specific neuron. And we have a series of empirical experiments. I'm happy to provide the link to the paper, where we show that this actually works in practice, where you can actually control the behavior of the network. You can sort of swap the neurons, and then the behavior of the network changes based on the swap. Now, the big unsolved question yet is, this assumes that what you're trying to encode is predefined. And if you think about it, it's clearly, this is not going to solve language understanding, but it's useful in many domains, including the biomedical space, where the ontologies are predefined, right? We know more or less, we know the proteins that exist in a body, in a species. We know the type of biochemical reactions we're trying to encode. So that ontology is more or less predefined. So we could encode that, right? Of course, it doesn't solve language in general, because language involves infinitely, right? So things change, things get added and change all the time. So it doesn't solve that problem. So that's what we're trying to do in that proposal. What we're trying to do, sort of ontology acquisition coupled with this training process, so the LLM adapts over time.
Thank you. I also have another question for myself, right? So,
Sure.
so when you apply those symbolic rules, right? So,
Mhm.
I wonder whether you have some treatment for those sentences or paragraphs actually they conflict, right, with that rule, right? So it does
Mhm.
not support, really does not support that rule, right? So
Yeah.
what's the treatment for those kind of
That's another great question. I love these questions. So this may happen for several reasons. So it could be that you can extract contradicting statements that are correct to contradict each other. And this may happen because biology is not the same between different species. So you may have sort of contradicting mechanisms between different species. You may have just bad science, right? So you can have a paper that's not really very rigorous. I would argue that you can detect that. So for example, we already have the building blocks for that. A good rigorous paper will have statistical analysis. So if your machine reading system detects that this paper did not carry a significance test, did not report the number of participants, etc., then that's not a very rigorous experiment. So you could measure the factuality of a paper using this type of information, including external signals, right? You can see how much a paper is cited, where is it published, and the design of their own experiments. So you can sort of find out information that's not supported by the facts and discard that. That's outside the scope of what I talked about today, but it is doable. And sometimes contradictions are fine, right? They come from
Yeah,
different
yeah.
species or the
So,
evolution of biology and so on.
Yeah, that's actually I have just had a follow-up question. Like, when that happens, right? So I wonder whether you have tried to actually try to retrieve additional evidence that actually to try to understand why they contradict. Because sometimes it's really maybe not in the same sentence, not even in the same paragraph, but somewhere else in that paper, right? They talk about a specific conditions and experimental settings and which they observe this contradicting
Right, exactly. Yeah.
things. So
So
that that important details, right, sometimes um, was not captured if you just naively apply whatever LLM or event extraction models to try to identify all those the entities, right? So and relations that might not, because that's a very fine detailed but important details, right? That kind of quantifies the under which condition that statement is true or not. Otherwise, it may not be
yeah,
true, right? So
I completely, sorry, go ahead. Sorry.
so I was wondering, like, do you have a plan to like, expand your system, right? To try to capture those important details that help to explain that what's seeming to be contradicting, but maybe nobody is wrong. It's just that they are talking about two different conditions.
Yeah, that's a very good point. In fact, this happens frequently because biology is not a linear process, right? You can't just say, if I'm having this protein, something is going to increase forever, right? And
Yeah.
in fact, yesterday, a student came to me, the student who's working on the LLM reasoning, he came to me with a single sentence where there were apparent contradictions. And the sentence was something along the lines, if we use this quantity of this protein, there's a downregulation. If we increase it by this much, it switches to an upregulation in the same sentence. So in one sentence was inhibition, the other part was promotion for the same protein and the same process. So it's exactly what you're saying, right? Depending on the conditions of the experiment, this changes. So we have sort of partial work on this in the sense that we do extract some conditions. So the conditions we extract are, what organism are we talking about? What organ are we talking about? What type of cells are we talking about? What we haven't done yet is what you just mentioned, which is sort of quantities, because that's obviously important. So we didn't get that far yet. So I would say we have a partial solution to that.
Great. Thank you. Yeah, I'm now handing back to Dr. Pei Ping.
Thank you, Professor Wei Wang. Again, congratulations, Professor Mihai Surdeanu. We are honored to have hosted your lecture. And for those of you who may have missed the lecture, I know there's a concurrent big meetings going on at NIH today. I think Jack is going to let all of us know where the recordings will be hosted and where the recordings will be available. My apologies to those of you who still have questions, but maybe not this time. We'll forward the questions to our speaker, and we can chat about these things offline. Back to you, Jack.
Yeah, thank you, Dr. Peng, and thank you, Mihai, for a great presentation. You have gotten us thinking a great deal about this interesting approach to LLMs. Neuro-symbolic models are very interesting, and machine learning, machine reading with synaptic patterns that carry semantic meaning is definitely fascinating. I will let everybody know who's still here that the lecture will be available on YouTube and on our training Vimeo. Please use the marketing materials or the beginning of this recording. You can see the link there. Thank you all for attending. We appreciate your contributions with this Q&A. And again, thank you, Dr. Mihai Surdeanu.
Thank you, Jack, and thank you all. This was fun. I enjoyed this, and thank you again for the invitation. I really appreciate it.